Okay, so what we talked about in the last podcast was what the significance of what Mendel did and how he, particularly how he did it, that was so significant with his excellent experimental design, and then the nature of his crosses, how he did his crosses, and what he saw. And we also talked about how what he saw related to the chromosomes, which he had no idea about. That's kind of what makes his ability to discern what he did so impressive. He had no idea about chromosomes or meiosis or any of that because he didn't know they existed. Yet he was still able to discern that each parent must contribute one gene, one allele per trait, and they must combine in these ways that we see in these monohybrid crosses. So what we're going to do now is look at a particular question that sometimes comes up because of the way these traits can be masked in this dominant recessive relationship. Two organisms can have the same phenotype but have different genotypes. For example, you can have a purple plant. Purple phenotypically be big P, genotypically its genes are big, okay? But you could also have a plant that is purple, but it's got a different genotype, big P, little p. Both of them look exactly the same. How can you tell them apart? How can you determine if an individual is pure breeding or not? Well, what you can do is something called a test cross. Okay? You take that dominant phenotype, this thing that looks, for example, purple, and you're like, I don't know what you are genotypically because I can't tell by looking, but you're going to cross it with an organism of a known genotype. We always know that the white must be little p because that's the only result that can give you white. If you're this, you're going to look purple. So you can't be that. You've got to be this. Okay? So you cross it with a known organism. Okay? This is called a test cross. When you breed something that has an unknown genotype, this dominant phenotype, which is you can't know for certain if it's big P or big P little b, and you cross it with something that you do know for certain. Okay? So a test cross works like this. If you have a dominant, completely dominant, pure breeding plant and you cross it with a recessive plant, you're going to get some different results than if you cross a heterozygote with a recessive plant. Okay? Which are you? Well, we can tell because if we cross the recessive with a pure breeding dominant, how are the offspring going to turn out? They're going to turn out all purple. But if you cross a heterozygote with the pure breeding white, then what you're going to see is that you're going to end up with at least half the time getting white offspring. And that way you'll know that this parent had to be a heterozygote, because always this parent is going to send little p's, but half the time this parent will send a big P, but the other half it will send a little p. So you end up with white, purple, purple. So your offspring, if you have 100 babies, roughly 50 of them should be purple and roughly the other 50 should be white, which is a 1-to-1 phenotypic ratio, whereas this will be a 1-to-0. There's nothing but purple over here. Okay? So that's a test cross. And now this brings us to one of Mendel's laws that he gave us, his first law of heredity, the law of segregation. This law basically says that each parent has two alleles. This parent has two alleles. This parent has two alleles. Even a parent that looks like this has two alleles. But they are only allowed to send one to the offspring, the alleles must separate or segregate, and we know that happens during meiosis. Okay? He didn't know it happened during meiosis, but we do. The alleles segregate or separate into separate sex cells, into separate gametes. Okay? And this is because homologous chromosomes separate from each other. Okay? You've got to remember that these genes, these alleles, are riding on chromosomes. So big P is riding on one chromosome, big P is riding on the other. Meiosis sees that these homologous pairs separate. This goes into one sperm, this goes into another sperm. Which sperm fertilizes the egg? We don't know. Okay? In this case, it wouldn't matter because they're both the same anyway. Same goes here. The only time where it gets a little interesting is if we have a heterozygote, because then when these separate, little p could go and fertilize the egg, or big P could. And again, we have no control over which one does. It's random. So this is the critical step. Meiosis, when the homologous pairs separate. If this has got big P, remember these chromatids are identical, so this would also have big P. If this had little p, and this one would also have little p, this one would go into one sex cell, a sperm. This one would go into a different sex cell. Which one's going to go and fertilize the egg? We don't know. They separated and we have no control over which one goes and fertilizes the egg beyond that. Okay? Now, so the law of segregation, you should know this. Which stage of meiosis creates that law? It is metaphase, essentially metaphase 1 into anaphase 1, where these homologous pairs separate. Okay? The homologous pairs separate. And Mendel predicted that even though he never ever saw it. Pretty cool. Okay? So, many of Mendel's experiments focused on just one trait. Flower color, pink versus white, one trait. Seed color, one trait, nothing else. Everything else about those plants would be the same except for the color of seeds they made. These are referred to as mono, one, hybrid, difference, crosses. Monohybrid, one trait. Well, we know that organisms are not that simple. There are many traits that can vary. So let's get at least a little more complicated and talk about crossing two traits that vary. A dihybrid cross. Okay? Let's make sure we understand this. Let's consider following two traits at the same time this time. Seed color and seed shape. Wrinkly versus smooth and round, or if you will, smooth, round, and yellow versus green. Okay? Okay. Let's look at a dihybrid cross. Let's say we are going to pick a plant that no matter how many times we let it self-fertilize, always produces offspring that produce yellow round peas. Always. The offspring always produce peas that look like that. Whereas we are going to cross it with a plant that is also true breeding, but when it reproduces, it always produces peas, and its offspring always produce peas that are green and wrinkled. We never see any other kind of pea. They're true breeding. So, looking at these, since they're true breeding, and this is true breeding, which one of these is dominant? Well, we don't know. Let's cross them and see what we find out. Well, we cross them, or Mendel crossed them, and what he found is that his F1s always turned out yellow and round. Well, now we say, okay, well, these traits must be dominant. If that's the case, what kind of notation are we going to use? Well, if yellow is dominant, then we're going to say big Y is yellow, and little y is going to stand then for green. If round is dominant to wrinkled, because that's what shows up in my F1, then we're going to use big R for round, and little r is going to be wrinkled. Okay? And so since everyone has to have 2 alleles per trait, then with regard to color, this yellow round pea plant must be big Y with regard to color of peas, but it also must be big R with regard to shape. We're following 2 traits now. This is trait 1 and this is trait 2. Over here, it must be little and little. Again, we're accounting for 2 traits now, okay? And they're both pure breeding, okay? Now, what do? We're going to cross these with themselves. We're going to allow these pea plants that are making these peas to self-fertilize. Oh, I do need to talk with you about what must be the genotype of all of these yellow round plants. Well, if this is Dad, let's say he's going to make sperm. What can he put in his sperm? Well, we consider this trait separate from this one. So he can send a big Y to his sperm. That's all he's got. He can send a big R to his sperm. That's all he's got. So all he can produce are sperm that look like that. Mom, all she can do is produce eggs that look like that, which means the babies must all be this, because when the sperm meets this egg, you're going to have big little. Okay? Now, let's let these plants self-fertilize. Let's let these plants grow up, produce flowers, and self-fertilize, and then plant the offspring, and let's see what we get. Okay? What Mendel noticed that he got was that he got, if he let thousands of babies be born from this cross, 9/16 of them would be yellow and round, 3/16 would be green and round, 3/16 would be yellow and wrinkled, 1/16 would show the complete recessiveness. Okay? The ratio he would get is a 9 to 3 to 3 to 1 ratio. Now, what explains that? What explains it is that the genes for these traits are on different chromosomes, and those chromosomes can sort out in meiosis in any way they wish. Okay? What this means is that do these genes have to travel together? Is that the result we get? In other words, does big Y always have to go with big R? Does little y always have to go with big R? Or can so the question is, what's going on? Okay? Let's think about what's happening at meiosis. Okay? We have a chromosome, perhaps, that has big Y. That means the other chromosome in the F1 has a little y. Okay? And then we have a chromosome that's got the big R in the F1 and one that's got the little r. Okay, so this is our heterozygote genotype, and we're just showing those on the chromosomes. But remember what happens during meiosis. All right, during meiosis, the chromosomes will line up. Okay, one member. If this is my cell at meiosis, one member of each homologous pair will line up on either side of the center of the cell. So big Y here, the chromosome with little y on it might be over here, the chromosome with big R might be here, chromosome with little r might be there. But is that the way they have to always line up? Could they line up like this with maybe big Y here and little y here, but with little r over here and big R over here. Could they? Yes. There's no law that says this chromosome has to travel over onto this side of the cell at metaphase that this one does, or that this has to go with this. They could just as easily align like that. They can assort themselves independently during meiosis. They are independent of each other. They can go into whichever side of that imaginary line that they want. That's huge, because that accounts for the hereditary pattern we see. We don't see that big Y, for example, always has to go with big R, or that little always has to go with little. What we see is that you can get these various combinations of gametes produced. Okay? Let's look. If this were the way the cells had to divide, the alleles had to travel, they had to travel like this always, then what we would see when the organisms reproduced is we would see a ratio that was essentially 3:1, round yellow to wrinkled green. We don't see that. What we see, what Mendel saw, is this result here. That's what we see. We don't see this result over here. What we see is that mom could make 4 different types of gametes. Remember, these are our F1 parents here. That's the F1 and that's an F1. They're both genetically the same. Dad could make all of these kinds of gametes. Okay? Because of that, we get all this variety in the offspring possible. Okay? That means look at all the different variety that can occur. We could have 9, if you'll count them, 1, 2, 3, 4, 5, 6, 7, 8, 9 that are round and yellow, 9. We could have 3 that are yellow and wrinkled. There's our 3 that are yellow and wrinkled. There's 3 that are green and round, and 1 that is completely recessive. That is what accounts for what we see. Okay? So Mendel's second law of heredity then is the law of independent assortment. So not only do the homologous chromosomes separate, but they can go in any combination that they wish when you're looking at more than one trait. Okay? Different loci separate into gametes independently. Okay? Round does not always have to go with yellow. It can also go with green, for example. Okay? And that's what we see. Okay, now the place where independent assortment is created in meiosis, because again meiosis is at the heart of this, is again in metaphase 1. When those chromosomes line up, they can line up like this, or they could just as easily have had the red on this side and the blue on the other. Okay? The exception, however, there is an exception to this law of independent assortment, and that is, remember, a chromosome has lots of genes on it. And suppose instead of being on separate chromosomes, the genes for pea color and shape were on the same chromosome. Then whatever they were would always travel together on the same chromosome. This is referred to as being linked. Okay? So if the genes travel together on the same chromosome, then that gives you a different result. They are said to be linked. Okay? So, law of segregation, it's not just associated with monohybrid crosses, but that's where we first start talking about it. Each allele segregates into separate gametes. Okay? The law of independent assortment says that when we are watching two traits or more at once, those traits can assort themselves in any combination they choose into gametes. It's independent assortment. Okay, and the exception is when genes are linked on to the same chromosome. Okay, we've already talked about that. Talked about that. All right, so that's it. Thanks for watching, and I'll catch you in the next podcast.